So we will now discuss about the eukaryotic gene expression and regulation. Now, gene expression in eukaryotes is regulated for what purpose? It is regulated to provide the appropriate response to the biological needs. What is needed by the cell? What is the time which is in? What is the time which is important now? Which protein is important now to form, and which protein is not required to form now? That will be regulated by certain genes, certain enzymatic, certain modifications that will silence that particular gene which is not required now to make protein, and they will also use some signaling molecules to make that particular gene express also for according to the needs of the cell. So this gene expression in eukaryotes can be occurred in the following ways. One, expression of certain genes which are also called housekeeping genes in most of the cell. So most of the cell have some common genes which are needed to express fully every time, all the time, that are called housekeeping genes. Then comes activation of selected genes upon demand. According to the needs, certain genes will be selected. And last, permanent inactivation of several genes in all but a few types. That means few cells will have that genes to be activated, but other cells they will not activate that particular gene. For example, your insulin is produced only by the beta cells of pancreas, and we know that this insulin gene can be obtained also from every cell also, okay, but it will be suppressed. So that information is suppressed in other cells. It is only expressed in that beta cells of pancreas. So these are the three ways through which eukaryotic gene expression can be done. First, the housekeeping genes will be all the time active in the cells, in every cells, whether it is your cells found in the liver, whether it is cells found in your lungs. They will express certain genes which are common in all of them. Secondly, activation of selected genes upon demand. To activate selected genes upon demand when we required, when the cell required that particular protein to be formed, then only that gene will be activated. And third is permanent inactivation of several genes in all but a few types. Best example I have said about the insulin. Insulin gene is found in every cells, but it is only expressed in the beta cells of pancreas. Similarly, glucagon, they are also an example. Growth hormone, it is also an example. Now, in case of prokaryotic cells, most of the DNA is organized into genes which can be transcribed and translated into proteins. In prokaryotic cells, most of the DNA is organized into genes which will be transcribed, which will be translated to form proteins. If you compare with mammals, very little of the total DNA is organized into genes. Very little. There is a very few percentage of your essential DNA which will be further organized into genes and their associated regulatory sequence which will regulate the expression of those genes. Why so much bulk of extra DNA is present? Still, it has not been observed. Still, it has not been studied till now. So, one important thing is that there may be, due to the expression, regulation of that genes maybe. So, eukaryotic gene expression and regulation are very highly complex. It is not like in prokaryotes we have talked about the lac operon and tryptophan operon, not just like simple. It is very complicated, and at different levels they will be regulating that particular gene. So, eukaryotic cells can regulate the expression at many levels, at many different levels. So, let us talk about these levels. Okay, so your first level is your epigenetic regulation. Epigenetic regulation related to your histone modification and chromatin structure. You have studied about in genetics about the nucleosome model, right? And that nucleosome is formed from the histone proteins, right? There were histone and non-histone proteins associated with the DNA to form the nucleosome. Okay, so this histone have a tail. It has a tail at its end terminal end, as you will see it later on. This end terminal tail is often modified by different functional groups, uh, methylation, acetylation, acetyl group, methyl group, right? Different phosphorylation is the common thing, and depending on at which position of this n terminal tail of histone is getting modified by this functional group, depending on that they will unwind or rewind, uncoil or recoil that particular DNA. Okay, so epigenetic regulation is actually the first level of regulation. Okay, because if the nucleosomes are very compactly, the histones are tightly compact that DNA to form that nucleosome, then DNA replication or DNA replication machinery will not come able to access that region. So it will be completely inactive. Not only DNA replication machinery, transcription machinery will also not able to access that region. But due to some modifications, if that nucleosome uncoil, DNA get uncoiled, then what happen? Then that region will be available for the transcription machinery to access that region and carry out their transcription. So expression of the genes occur. So this is the first level of expression, epigenetic regulation, which is your histone modification and chromatin structure. Then comes your enhancers and tissue specific gene expression and repressors. There are some enhancers which will allow the gene to be expressed. This works in the level of transcription, right? And there are some repressors also which will do the opposite thing of enhancer. They will repress that particular region. They will not allow that gene to be transcribed. Third is your RNA splicing, including your alternative splicing. This is also one more important thing. The splicing chapter that I have taught to you before. This splicing also is a type of regulatory mechanism done by the eukaryotes to express that gene or not. Okay. Then comes the fourth level of expression. So at different levels the eukaryotic gene is regulating. See, so much complicated. The fourth one is control of RNA stability. If you can able to see it, let us see. So control of RNA stability like UTR. and the microRNA, also called miRNA. Then comes the fifth one, motifs in protein and gene expression. Motifs are special structures of the protein. These motifs will help to interact with the DNA. Most of the transcription factors are having this type of motifs. Now motifs are of many kinds, like helix coil helix motif, leucine zipper motif, zinc finger motif. Okay, the name is you are you will see that these are not so much difficult. Actually, these motifs are certain special structures found in the proteins which will help to interact with the DNA for regulating that DNA, okay, to make that DNA, that particular gene, on or off. Okay, last is your two more things are there. Number 6, processing of the mRNA. It is very important. Without processing, you cannot able to use that mRNA for protein formation. Processing, you know very well, the splicing, the 5 prime cap formation, the poly A tail formation, right? And the lastly that I have already taught to you is number 7, which is your UORF. In the translation regulation can also be done for this eukaryotic gene expression and regulation. UORF I have already taught to you. They are the upstream open reading frames which are just located before the main open reading frames, and depending on the situations they will not allow that mRNA to be translated to form the protein, right? Best example is GCN2 that I have talked to you. Okay, now these are the levels of eukaryotic cell gene expression. Now today I will talk to you only about the epigenetic regulation because it is quite lengthy and it will take a long duration of the video, which will be very too much of storage. It will hamper your storage. So let's move on to the epigenetic regulation. Okay, so we have come to this epigenetic regulation, histone modification and chromatin structure. So what they are saying, the DNA in the nucleus is precisely wound, folded and compacted into chromatin. Wait for a minute, please wait for a minute. Okay, so it is saying that the DNA in the nucleus is precisely wounded, means coiled, folded and compacted with the help of histone and non-histone proteins. So here a little bit small I have written. So the DNA in the nucleus is precisely wound, folded and compacted with the help of histones and non-histone proteins into chromatin. Chromatin are actually protein DNA duplex complex which will be further condensed. This chromatin will be further condensed into chromosomes so that it fit inside the nucleus, right? So DNA in the nucleus is coiled by these histone proteins to form the chromatin structure. This chromatin structure will be further condensed into chromosome so that they will fit inside the nucleus. Remember, chromatin are of two types. From class 12 you have learned euchromatin and heterochromatin. Heterochromatin are tightly coiled chromatin which are your chromosomes, and euchromatin are the loosely coiled chromatin where your DNA repair, where your DNA replication machinery, transcription machinery can able to access. So this chromatin is organized. This chromatin is organized so that specific segments can be accessed as need by a specific cell type. Why this chromatin is organized? This chromatin is organized so that specific segments can be accessed as needed by specific cell type. Means specific cell, if it requires, if it wants to use that particular region of chromatin, then it will, with the help of some modifications like I have said, by attaching acetyl group to the N terminal tail of histones, or by transferring the methyl group. Okay, in this way they regulate the specific gene segment. So the first level of organization or packing is winding or coiling of the DNA strands around the histone proteins to form nucleosome. So this is the first level of organization, this histone modification and chromatin structure. It is your packing or winding, means coiling of DNA strands around the histone proteins to form nucleosome. Okay, under electron microscope, this coiling of DNA around the histone proteins to form nucleosome looks like beads on a string. Obviously, we have already discussed in the genetics that in electron microscope, the coiling of DNA around the histone protein to form nucleosome looks like beads on a string. Here is your structure of a typical nucleosome. I tried to draw in a three-dimensional view. So see, this red, the pink one is your DNA. 146 base pair of DNA is required to wrap around this histone octamer. Histone octamer means 8 histone proteins are involved. There are usually 4 types of histone proteins. One is H2A, H2B, H3 and H4. So each of these types of histone protein are found 2 molecules. Okay, so they are the core histones, right? So if DNA encoding a specific gene is to be transcribed, suppose the cell wants to transcribe that particular DNA which is wrapped in this nucleosome, right? Then the nucleosome surrounding that region of the DNA has to unwind. It has to uncoil now. Okay, to open that specific chromosomal region and allows for the transcriptional machinery, that is your RNA polymerase, to initiate the transcription. So suppose a particular region of that chromatin is required by the cell, that yes, we want to transcribe this region, we want to express that gene into protein. So that only that region of the DNA will be unwinded, from the nucleosome by some modifications in the tail of these histones that I have shown to you. So these are the tails, the N terminal tails of the nucleosome of these histones, right? Okay, and these tails, as histones are protein, it is made up of amino acids. Actually, some functional groups are attached to these tails at specific amino acid, and depending upon that which amino acid has been modified by which functional group, depending on that, they will change their conformation and allows that DNA to unwind or rewind. Okay, so this is the main thing about this, the first level of gene regulation and expression in eukaryotes. Suppose if a DNA encoding a specific gene to be transcribed into mRNA, then nucleosomes surrounding that region of the DNA has to uncoil. Okay, has to unwind, because it is tightly packed with the with the histone to form chromatin. So how will other proteins will access that region? So it has to unwind, so that DNA will open that specific chromosomal region and allows for the transcriptional machinery to carry out transcription and later on translation process. Okay, so this was about the epigenetic regulation. More to come, let us see. So nucleosomes can move to open the chromosome structure and to expose a segment of DNA, but they do so in a very controlled manner. It is not like that nucleosome opens automatically. Okay, it has to be modified. So this nucleosome can move. This nucleosome can move to open the chromosome structure, um, or to expose a segment of DNA, but they do so in a very controlled manner. How they do control, let us see. As I have mentioned just now, there are n terminal tails present on the histone octamer. Octa means 8, so there are n terminal tails at the amino terminal end, which we in short we call n terminal tail. Okay, amino acids proteins always start from amino terminal end and ends at carboxy terminal end, that you know. So there are n terminal tails present on the histone octamer. These are the histones which form that histone octamer: H2A, H2B, H3, H4. Basically, two molecules of H2A, two molecules of H2B, two molecules of H3, and two molecules of H4 are needed to form this histone octamer, and all of them have a n terminal tail, right? This n terminal tail has been protrude means it is found, it is protruding out, means it is found outside from the histone core of the nucleosome. As I have said, look at this figure. So it has been shown that these histone tails are found outside the histone octamer. See, H2A have this n terminal tail, H2B have this n terminal tail, H3 have this n terminal tail, and H4 also have this n terminal tail. So they all protrude outside the histone octamer. Okay, so they grow outside from the histone octamer, this n terminal tail from the core of the nucleosome. Now this tail is actually modified. Okay, modification of the tail, modification of the tail of the histone by various chemicals like your acetyl group, by methyl group, by phosphate is a common for phosphate group, can allow the gene regulation. Modification of this tail at certain region of this tail of this histone tail is done by various chemicals. And what are those chemicals? Like acetyl group, methyl group, phosphate group can what? Allows the gene regulation. How? By binding that chemical group, by binding this acetyl methyl phosphate group to the tail at certain regions, they will, it will change its conformation and allows coiling or uncoiling of the DNA of the specific region of the chromatin. So modification of the tail of the histone by some specific functional groups like your acetyl group, methyl group, phosphate group can helps gene regulation by coiling or uncoiling the DNA of the specific region of chromatin. So this was the basics of your histone modification. Now we will just talk about some specific groups which causes this uncoiling or coiling of the histone protein, so that the nucleosome get unwinded, the DNA get unwinded from the nucleosome and carry out the expression, or it will rewind. Regulation means two things: you have to unwind that particular region, or no, we don't want that protein, we don't want that protein to be formed. So the gene which is responsible for that protein has to be rewinded to form a heterochromatin. So for that also we use certain functional groups also. The first thing is your acetylation and deacetylation. So histone acetylation and deacetylation, this is a basic thing. Histone acetylation and deacetylation are the processes by which the lysine residues within the N-terminal tail of histone core of nucleosome are acetylated. Again I repeat, histone acetylation and deacetylation are actually what? They are the process by which the lysine amino acid within the N-terminal tail of the histone core of nucleosome are acetylated. So histone acetylation and deacetylation means the process through which your lysine residues within the N-terminal tail of the histone will be acetylated or deacetylated. Means what? Acetylation means acetyl group added within lysine amino acid. I have written in short form LYS. Acetyl group added within lysine amino acid residues within N-terminal tail of histone octamer. And deacetylation is the opposite of acetylation. Removal of acetyl group from the N-terminal tail of histone octamer. It is written in short, very small way, so I hope you can able to see. So see what it says: removal of acetyl group from N-terminal tail of histone octamer. Okay, so this acetylation and deacetylation are the basics of chromatin modification, okay, regulation. So this acetylation and deacetylation are actually a part of gene regulation, right? So acetylation simply means transfer or addition of a acetyl group where to the lysine residues found within the N-terminal tail of histone octamer, and deacetylation means removal of that acetyl group from that removal of that acetyl group from that N-terminal tail of histone octamer. These are the part of gene regulation. Now normally acetylation predominantly occurs on the lysine residues. See, acetylation predominantly occurs on lysine residues in the amino terminal ends, that is your N terminal ends of histones. So this acetylation normally occurs on the lysine residues in the amino terminal ends, that is your N terminal ends of your histones. This modification in histones reduce the positive charge of the terminal ends. Let us see why it. This is the main important thing. So it is saying that acetylation predominantly occurs on the lysine residues. It mainly occurs in the lysine residues that are found on the amino terminal ends of histones. What happens when acetylation occurs on these lysine residues of N-terminal tail of histones? This modification in histones actually reduce the positive charge of the terminal ends. The terminal ends have a positive charge. Due to addition of this acetyl group on lysine residues, it reduces the positive charge and decreases the binding affinity to the negative charge DNA. Lysine and arginine are positive charge amino acid. I may be right. Okay, for verification just look at the internet and see, but I am sure mostly that lysine and arginine are positively charged amino acids. They are found on this N-terminal tail of the histones, and due to the effect of this lysine and arginine, they can able to bind to that negative charge DNA. DNA is negative charge, you all also know, because it has a phosphate group on its backbone, right? So this lysine and arginine, which are actually found on the N-terminal tails of the histone, it makes that DNA, it helps to interact with the DNA at a high affinity with the DNA to bind. But suppose if you reduce the positive charge of these histones, how? By binding the acetyl groups, by binding acetyl groups to the lysine residues of the histone, then what happen? It will decrease the binding affinity with the negative charge DNA. Consequently the nucleosome structure will be disrupted to allow transcription of that particular segment. This is written segment, this one is segment at the corner, to allow transcription of that say particular segment of DNA. The simple thing, there are lysine residues, there are arginine residues found on the histone tails. There are also other amino acid residues we'll talk on later. Acetylation normally occur on lysine residues, means acetyl group is normally added to the lysine residues of the N-terminal tail of histone. Now when acetylation occur on these lysine residues of this histone tail, then what happen? There will be reduced binding affinity to bind with the DNA, because DNA is negative charge, and with the help of these positive charge amino acids, these histone bind with the DNA, and this most of the positive charge amino acids of the histone found at the tail. One of the positive charge amino acid is your lysine, then arginine, right? So when this lysine get acetylated by binding acetyl groups, what happen? It will reduce that positive charge of terminal ends, that is the tailgate has its reduced positive charge now, and decreases that binding affinity to the negative charged DNA. Consequently the nucleosome structure is disrupted and allow transcription of that particular segment of DNA. So this was the thing about the histone acetylation. Now we have to say that this histone acetyl group does not bind automatically, obviously. It is catalyzed by some enzymes mainly, and that name of the enzyme is your this one, HAT. Histone acetylation is catalyzed by the enzyme called HAT. It is short form. The full form is histone acetyl transferase. Histone acetylation normally catalyzed by this enzyme called histone acetyl transferase. This HAT enzyme catalyzes the transfer of acetyl group. This HAT enzyme catalyzes the transfer of acetyl group, which is your CH3CO, from one molecule, that is from acetyl coenzyme A. It transferred that acetyl group C. From acetyl coenzyme A, it transferred that acetyl group to another. Here it transferred that acetyl group from acetyl coenzyme A to the N-terminal tail of histone, particularly at the lysine residues. So histone acetylation catalyzed by histone acetyl transferase. Again I repeat, histone acetylation is catalyzed by the enzyme histone acetyl transferase, and this histone acetyl transferase can catalyze transfer of the acetyl group, that is your CH3CO. This acetyl group is transferred from acetyl coenzyme A to the N-terminal tail of histones at lysine residues, right? Okay, the main function of HAT is only to transfer the acetyl group from acetyl coenzyme A. Here it is transferring the acetyl coenzyme A to your N-terminal tail of histones, particularly at the lysine residues. Now comes about the deacetylation. It is the opposite of acetylation. It is the removal of the acetyl group from the N-terminal tail of histone, allow that histone to again coil that DNA to have a high affinity then to form in the histone octamer protein. So deacetylation is simply the reverse of the acetylation reaction, where your acetyl group will be removed from the N-terminal tail and allowing the to coil the DNA in the histone octamer protein. Now here histone deacetylation is also catalyzed by another enzyme called histone deacetylase. Histone deacetylase, here you can see the name. This histone deacetylation is carried out by this enzyme histone deacetylase, and histone deacetylase is denoted by a short name called HDAC. H for histone, DAC for deacetylase. Now acetylation of histones, actually I have already mentioned, just we have repeating here. Acetylation of histones remove the positive charge of the histones, thereby decreasing the interaction of the negative N-terminal of histones. Due to acetylation of histones, it removes the positive charge of the histone and decreases the interaction of the N-terminal histones with the negative charge phosphate groups. The negative charge phosphate groups found in the DNA, and when histones are acetylated, the positive charge of the histones which are found in the tail due to the presence of lysine and arginine like positive charge amino acid residues, if these get acetylated, then the interaction of the N-terminal tail with the negative charge phosphate group will be decreased. As a consequence, the condensed chromatin is transformed into more relaxed structure. Now the interaction become weak now between the histones and the DNA, because the histones positive charge is getting reduced by transfer of acetyl groups to that positive charge amino acid, and thus the interaction between the histones with the DNA getting decreased. Thus as a consequence, condensed chromatin, which is also called as heterochromatin, right? So the condensed chromatin is now transformed into more relaxed structure called euchromatin now, and that is associated with greater levels of gene transcription, right? And this process can be again reversed by the help of histone deacetylase as denoted by HDAC. So it is written that this relaxation can be again reversed. It can be again reversed to form condensed chromatin called heterochromatin by the HDAC activity. That is, with the help of histone deacetylase, you can again convert that euchromatin into heterochromatin form. Here some terms has been given. Relaxed transcriptionally active DNA is referred to as euchromatin. Relaxed transcriptionally active, but those chromatin which actually can access by the transcriptional machinery to do the transcription is called transcriptionally active DNA. Those chromatin where transcriptionally active DNA can be occurred are there is called euchromatin, where more condensed or tightly coiled chromatin is referred to as heterochromatin. Condensation of chromatin can be brought about by the process of deacetylation and methylation also. This condensation means from euchromatin to heterochromatin. This can be brought about by deacetylation as carried out by deacetylase enzyme, histone deacetylase, and by methylation also condensation of chromatin can also be done. Okay, now methylation is little bit different. It has no it has no effect with the charge actually. Methylation has no effect with the charge, that methylation occurs at particular site. Methylation means addition of methyl groups, you know. So methylation can also cause condensation of the chromatin. That means it will form heterochromatin, but it has no acting upon charge because methyl group is also a positive charge. Okay, right? So we will now move on to the next page. Now we will talk about, this was about the histone acetylation and deacetylation. Now I will talk about important thing of histone methylation. It is very important, and it should be studied thoroughly, and it should be done with properly. Okay, now about histone methylation. Histone methylation is associated with many gene regulation. Okay, as you have seen in different ways. Let us see. So histone methylation is a process simply by which methyl groups are transferred to the amino acids of histone proteins that make up the nucleosome. Here histone methylation means your methyl groups will be transferred to the amino acids of histone proteins that makes up the nucleosomes, on which DNA double helix wrap around to form chromosomes. Simple thing, just like acetylation, histone methylation is a process by which methyl groups will be transferred to the amino acid. Which amino acid we will talk later on, of the histone protein. They will interact with the N-terminal tail, that's for sure, this histone methylation, and by transferring of methyl groups, that is your CH3 group, it will modify the nucleosome structure on which DNA double helix wrapped around again tightly to form chromosomes. Now methylation of histones can either increase or decrease the transcription of genes depending on which amino acids of the histones are methylated and how many methyl group has been attached. Methylation of histone only does not means that the chromosomes will be condensed, silenced totally, not like that. Depending on which amino acid of your histones are getting methylated and how many methyl groups are attached, depending on that, methylation of histone can either increase the rate of transcription or decrease the rate of transcription. Sometimes it can silence the transcription of that particular gene segment. So depending on which amino acids on the histones are getting methylated, depending on how many methyl groups are getting attached to that particular region of histone tail, depending on that, methylation of histones can cause increase in the rate of transcription or decrease in the rate of transcription. Okay, now the main basic thing is that methylation event that will weaken the chemical attraction between the histone tail and DNA will increase the rate of transcription. Again I repeat, methylation event that will cause weaken that chemical attraction between the histone tail and DNA will increase the rate of transcription. Those methylation that cause low affinity that will leads to low affinity between the histone tail and the DNA will cause increasing the rate of transcription, because they enable the DNA to uncoil now, so that the transcription factor proteins and RNA polymerase, which are the components of transcription machinery, they can able to access that DNA. Okay, so methylation of histones, as we will proceed through a good example then later on, little bit later on. So this process of methylation of histones is very critical for the regulation of gene expression that will allow different cells to express different genes, right? So this process of methylation of histones is very critical for regulation of gene expression, and it will allow different cells to express the different genes. Okay, in some of the events methylation can also cause a high affinity between the histone tails and DNA. In some case we can see that methylation weakens the chemical attraction between the histone tail and the DNA, depending on, I have already said, depending on which amino acids the in the histones are getting methylated, and how many methyl groups are attached. Depending on that, it will allows to uncoil that DNA from the nucleosome or recoil, so that transcription machinery can able to access that region during uncoiling. Okay, so this process of methylation is very critical for the regulation of gene expression, which will allow different cells to express different genes, right? Let us move on to the next page. Okay, one more thing, I have already said this, mentioned above, that histone methylation can be associated with either transcription repression or transcription activation. Either histone methylation will cause transcription to be permanently become inactive, or it may cause you activation. Now it is depending on which amino acid is getting methylated, which amino acid in the histone tail is getting methylated, and how many times, how many methyl groups is getting attached to that site. Depending on that, histone will cause you activation or repression of that transcription of that particular gene segment, okay, to coil or uncoil. Now comes the amino acid that are mostly getting methylated, right? It is saying that histone can be methylated on lysine, denoted by one letter code called K, and arginine, denoted by R residues. So histones can be methylated on lysine and arginine residues only, but methylation mainly commonly observed on lysine residues of the histone tail H3 and H4. So remember, histone can be methylated on lysine and arginine residues. Generally speaking, histone methylation or histones can be methylated on lysine and arginine residues, but methylation most commonly is observed on lysine. Methylation is most commonly observed on lysine residues of histone tails H3 and H4. Okay, now this has been shown that the basic thing that amino acids are always read from N terminal tail to C terminal tail. Amino acids always read from the amino terminal end and proceed to the carboxy terminal end. Just a little bit of simplicity for making you to recapitulate the basics. So normally the histone tails of H3 and H4. H3, H4 are the histone. They have also the tail. They also contribute to form nucleosome to form the histone octamer. So the histone tails of H3 and H4, these tails are often get methylated, done by methylation. Methylase, some methylase enzyme, okay. And basically methylation observed on lysine residues of this histone tail of H3 and H4. Now comes to the common sites of methylation with gene activation. This means methylation that leads to activation of the genes. This is a shortcut, you have to remember this. Methylation that is associated with activation of genes. Which are the sites? On which site of histone H3 and H4 when methyl groups attach will cause the DNA to unwind from the nucleosome and become activated? They are H3K4, H3K48 and H3K79. Now what does it means? H3 for histone H3, K refers to lysine, 4 refers to the fourth amino acid residue. So within the histone H3 of the histone octamer, the N-terminal tail of histone H3 have a lysine residue at 4th number position. If that get methylated, it will be associated with gene activation. That means DNA will unwind from the nucleosome so that transcription can occur. Another region is also there, H3K48. H3K48 means H3 for histone H3, K for lysine, 48 means the 48th amino acid is lysine found on histone H3, which is methylated, and that leads to gene activation. It leads to unwinding of the DNA, uncoiling of the DNA from the nucleosome to access, so that transcription machinery can access. Another region is also there, H3K79 means the lysine amino acid found on 79th region of histone H3. Now these are the common sites for gene activation. What are the common sites for gene inactivation? It is your H3K9 and H3K27. Means within the histone H3 there is a lysine at the number 9th position. This 9th position, when it gets methylated, it will leads to inactivation of the genes. It will again recoil, okay. And another region, H3K27. This is also another important region which will leads to inactivation. So this methylation of histone tails at different residues serves as a markers for recruitment of various proteins or protein complexes that are serving to regulating the chromatin activation or inactivation. So this methylation of histone tails at different residues, they serves as a marker. They are serving as a marker for recruitment of various proteins or protein complexes that will serve to regulate the chromatin activation or inactivation. So this is the basic thing of your histone methylation. I have just said that I will go to your example and some more important terms based on this, okay. So let us see if I can access that part, okay. So what is written? It little bit of haziness because of my problem with camera also. So DNA methylation is associated with silenced genes in mammalian cells. DNA methylation is associated with silenced genes in mammalian cells. So silenced gene means to inactivate the gene so that the gene will not be transcribed now. So some mammalian genes are kept silent by methylation of nearby DNA sequence. Some mammalian genes are kept silent by methylation of nearby DNA sequence. In fact, large regions of the mammalian genomes are marked. Marked means marked by methyl group in this way. Large regions of the mammalian genomes are marked by methylation by methyl groups in this way, and often DNA methylation is seen in the regions that are also heterochromatic. I have just now said that common sites for gene inactivation is H3K9 and H3K27. When that region is getting methylated, when the ninth position of the histone H3 that contains lysine, or when the 27th position of histone H3 containing lysine is getting methylated, it leads to heterochromatin formation, right? So it is saying like this, that some most of the regions of the mammalian large regions of the mammalian genomes are marked by methyl group in this way, and often DNA methylation is seen in regions that are heterochromatic. So methylation is seen in the regions that are normally heterochromatic. Okay, why? There are some proteins which are associated to form that heterochromatin. Okay, so this is because methylated sequence are often recognized by some other proteins, like some DNA binding proteins like MECP2, that will recruit histone deacetylase and histone methylase, which will modify near the chromatin. Thus, methylation of DNA can mark the sites where heterochromatin are subsequently forms. So again I repeat, some mammalian genes keep silent by methylation of nearby DNA sequence. Some mammalian genes are kept silent by methylation of a nearby DNA sequence. In fact, large regions of the mammalian genomes are marked by methyl group in this way, and often DNA methylation is seen in regions that are also heterochromatic. I have shown you that some important regions of the histone H3 containing lysine can be methylated to silence that region, to inactivate that region. Methylated sequences are often recognized by some DNA binding protein like MECP2. When these methylated regions are recognized by this type of DNA binding proteins, then what happen? It will recruit the histone deacetylase and histone methylase also, which will modify that chromatin. Thus, methylation of DNA can be marked sites where heterochromatin subsequently forms. This DNA methylation lies at the heart of a phenomenon called imprinting. There is a technical term that you might have heard during your genetics lecture: genomic imprinting. Genomic imprinting means it is predestined that which genes will be activated, which genes will be repressed. Okay, so DNA methylation lies at the heart of a phenomenon called imprinting. Okay, now in a diploid cell there are two copies. Now we are going deeply into the imprinting process, where where it will show that how methylation predetermined that this part of genes should be activated forever, this part of genes should be inactivated, right? Now in diploid cell, you know, 2n having two sets of chromosome, one from father, one set from mother, one set from father. So in a diploid cell there are two copies of most gene. One copy of gene of a chromosome is inherited from father and the other copy of the gene on the equivalent chromosome comes from the mother, right? In most cases these two alleles are expressed at comparable levels. In most cases these two alleles are expressed at comparable levels. It is hardly surprising that the chromosomes carry some regulatory sequence and are in the presence of same regulators. They are also located in the equivalent region of two very similar chromosomes. But there are few cases where one copy of a gene will be expressed where the other is silent. See, it is saying that suppose we get the two sets of chromosome from father and mother. For example, the chromosome number 1 we also get from father and we also get from mother. The chromosome number 1, I am giving an example. The chromosome number 1 consists of the genes, consists of the essential segments from the father that we get and from the mother we get. But surprisingly, we see that they also have regulatory sequence, and this chromosome also, the chromosome number 1 of from father also have some regulatory sequence, and chromosome number 1 from the mother also have some regulatory sequence. But mainly if we think the peculiar thing that one copy of the gene from the chromosome number 1 is expressed, while that same gene in the maternal chromosome has not been expressed, have been silenced. Why this is the case? On the other hand, in the maternal chromosome, one is expressed equivalent to that father's chromosome that has not been expressed. Same genes, they have a regulatory sequence, but both of them is not expressing, partially expressing, not partially. One is determined that this gene from the father will be expressed and this gene from the mother will be expressed, right? So in most cases two alleles are expressed at comparable levels. This is hardly surprising. Chromosomes carry same regulatory sequence and are in the presence of same regulators also. They have a regulation. By methylation they can be regulated. They are located in the equivalent region of two similar chromosomes, that is called locus. The regions of the chromosomes, the regions of the genes are called locus in the chromosome. But there are few cases where your one copy of a gene is expressed, the same other copy from the maternal chromosome may be silent. Why this is happening? That is your genomic imprinting, that is your gene imprinting, and that is done by methylation. Okay, so more to go. So to understand about this genomic imprinting and also to understand that how your methylation leads to inactivation of genes, two well-known studied examples are your human H19 gene and IGF2 gene. Okay, so IGF2 gene and H19 gene both are located in the human chromosome number 11, and a very well example to show this genomic imprinting is your H19 and IGF2 gene. I will go to the figure just in the next page, given in the next page. In a given cell, one copy of H19, let us see, we get that 11th number chromosome from father also and from mother also, right? In a given cell, one copy of H19, that is on the maternal chromosome, is expressed, while the other copy on the paternal chromosome is switched off. It has been observed that this H19 gene found on the maternal chromosome is expressed, while the H19 gene found on the paternal chromosome is switched off. For the IGF2, it is reverse. That is, the paternal gene that contains IGF2, the paternal chromosome number 11 that contains IGF2 is expressed, while the maternal chromosome number 11 which contains IGF2 gene is repressed. Why this occurs? So the paternal copy is on in case of IGF2, while the maternal copy is off in case of IGF2. While in case of H19, the maternal chromosome, chromosome number 11 containing H19 gene is on, means it is active, while paternal chromosome for the H19 gene is off. Now this is done by regulation, right? And also some other proteins. Here you comes your enhancer and insulator, right? So two regulatory sequences are critical for your differential expression of these genes. One is your enhancer, located at the downstream of the H19 gene, and the insulator, located between the H19 and IGF2 genes. So now you will not able to understand. For this we have to see the figure. Let us see the figure. Okay, so this is the figure. So this is the normal gene simplified diagram, which shows the H19 gene and your IGF2 gene. The first figure that you can see given as A, it is your maternal chromosome number 11. Okay, the maternal chromosome number 11 have this IGF2 gene in off position, while the H19 gene is in on position, right? Now here mainly speaking, enhancer, which has been shown in extreme right of your page in this picture, is regulating this type of expression. Okay, and methylation also plays an important role. We will see them just bit of second. So enhancer, which is located in the downstream of the H19 gene, and your insulator, which is located between the H19 and the IGF2 gene. See, the enhancer normally located between the enhancer is located at the downstream, on the downstream of the H19 gene, while the insulator, as given here, you can see able to this. Okay, this insulator region, see this segment is the insulator. There is a protein has been bind, that's another issue that I'll talk you later on. The insulator is located between IGF2 and H19. Okay, and enhancer is located in the downstream. Okay, so this enhancer, so this enhancer, which is located in the downstream of the H19 gene, and insulator is located between H19 and IGF2 gene. So when enhancer region, suppose when this enhancer region is bounded by activators, when this enhancer region is bounded by activators, it can activate either of the two genes. Again I repeat, when this enhancer region of the DNA is bounded by some activator enzymes, activator proteins, it will activate either H19 gene, or it can activate IGF2 gene. Let us see again to the figure. So it has been shown here, this enhancer region has been bounded by an activator. This activator can either allow the expression of the H19 gene, or it can allow the expression of IGF2 gene. How? So here two important things come into play. Okay, so when enhancer region is bounded by activators, again I repeat, when enhancer region bounded by activators, it can activate either of the two gene, right? But the main reason is, if the enhancer can activate either H19 or IGF2 gene, why does not it activate randomly? Why doesn't it activate randomly? That means, why it activate only H19 on the maternal chromosome, and why this activator activate only IGF2 on the paternal chromosome? Why not it activate the H19 on the paternal chromosome? Why not it activate the IGF2 gene on maternal chromosome? The answer lies in the role of insulator. I have shown you the insulator. See the figure, the insulator, right? See the figure of insulator. What has been shows the insulator? If you look at the picture, a protein has been bind, right? CTCF, a protein called CTCF binds to the insulator, that is preventing the enhancer to express the IGF2 gene, while it is allowing this H19 gene to be expressed. Okay. So we'll talk about this. The simplicity I am talking about, that CTSF bind to the insulator and preventing the enhancer, better to say preventing the activator which is binding to the enhancer to prevent the expression of this IGF2 gene, while it is allowing the expression of this H19 gene. This was in the case of maternal case, right? In the paternal case, we can see this is the B, the paternal chromosome. In the paternal case, we can see there is a methyl group attached to the insulator region up to the H19 region, that prevents the this CTCF to bind. Insulator there, your CTCF protein will not bind because of methylation of these regions, and thus now the activator which has been bind to the enhancer, it can now able to express the IGF2 gene. So this is the case in case of paternal and maternal chromosome number 11. That why maternal chromosome number 11 has only expressing this H19 gene, why not it is expressing the IGF2 gene? Because within the insulator a protein binds called CTCF, and what does CTCF protein means? Let us see. CTCF, let us see if it gives the name. The CTCF name has not been given here, sorry for that, we have to search it from the internet source. Okay, so the CTCF gene bind to the insulator and it is preventing the enhancer um, to activate this IGF2 gene, while it is allowing the CTCF gene bounded to the insulator, allowing the H19 gene to be turned on. This is in the case of maternal chromosome, while in the case of paternal chromosome, the insulator region up to the H19 region has been methylated, has been methylated, and due to methylation this CTCF protein cannot bind to the insulator. This is the case, that why this activator now can able to access to this IGF2 gene to turn on, while the H19 gene is turned off in case of paternal chromosome number 11, right? Okay. So it has been shown that um, so the insulator binds to a protein called CTCF that will block the activators at the enhancer region from activating the IGF2 gene. So CTCF is actually blocking the activators so that the activators cannot access or activate the IGF2 gene, while CTCF permits the expression of H19 gene on maternal chromosome. In the case of paternal chromosome, in contrast, insulator element and H19 promoter are methylated. Again I repeat, on the paternal chromosome the insulator element and H19 promoter are methylated. In that site transcription machinery cannot bind to the H19 promoter, right? Because the transcription machinery, because that region is methylated, transcription machinery cannot able to access. CTCF also cannot bind to this methylated insulator element, right? So due to methylation Due to methylation at the due to methylation at the insulator region and also at the H19 promoter region, the transcription machinery cannot able to bind, as well as your CTCF protein also cannot able to bind. As a result, enhancer can now activates the IGF2 gene. Okay, the H19 gene is further repressed on the paternal chromosome. Okay, it is further repressed by the paternal chromosome by the DNA binding protein called MECP2. There is another protein called MECP2 which binds to the H19 gene of paternal chromosome to repress, for more repression of the H19 gene. So this is, you can see here very well that how your regulation is done by methylation. Okay, you can see here very clearly, and this is a part of genomic imprinting. Okay, so this is the case, you can able to note it down, this writing. Okay, I will provide these images to you just after this lecture. So these patterns of gene expression must sometimes be inherited also. I have said just about the H19 and IGF2 gene. These patterns of gene expression may sometimes be inherited. Okay, and a signal will be released by one cell during development that will cause neighboring cells to switch off that specific gene. If you see in the case of zygotic development, during development of embryo, then there will be some signaling molecule released from one cell during development. It will cause the neighboring cell to switch off that particular genes, as in the case we see in maternal gene, their chromosome number 11, their IGF2 is been silenced due to that CTCF protein. So those genes may able to remain switched on in those cells for many cell generations, even if the signal that induce them present only for a short time. So this inheritance of gene expression patterns in the absence of either mutation. So these is a type of gene expression patterns from the developmental stage. It is inherited. That is called your epigenetic regulation. It is not like mutation. Okay, it has been predetermined. Okay, and this imprinting is the imprinting example discussed above reveals one way that expression of a gene can be regulated epigenetically, right? So this was about the methylation part. I will talk to you the next one later on in the next video lecture, that is your enhancer, tissue specific gene expression. There are many more to do, the motifs in the DNA, which are a very important part. Okay,